My name is Thea Newman and I'm the director of Solaravus. In this video, I'll be telling you about some ideas that I've been working on within Solaravus on the COVID-19 pandemic. Details of everything that I will be talking about can be found in the Solaravus technical report that has been written on this work. That is available from the Solaravus website, and there is a link in the description below the video that will take you directly to that paper. There is also a link to the Google Scholar profile for me, Thea Newman, just so that you can have more of an idea of my background. And I will just speak very briefly about that now, because the vast majority of viewers of this video will never have heard of me. So yes, my name is Thea Newman. I was trained in theoretical physics at the universities of Oxford and Manchester many years ago, and I have spent my career studying physics and biology using theoretical physics techniques. For the past 20 years, I've been working in a variety of areas in biology: ecology and evolution, developmental biology, genetics, molecular biology, and most recently, cancer. I have held academic positions and professorships in universities in the United States and in the United Kingdom, most recently working as Chair of Biophysics and Chair of Systems Biology at the University of Dundee. In 2018, I decided to set up an independent company, Solaravus, because I had a range of projects that I wanted to pursue that required me to be independent. I'm still very actively involved in science, and this particular project about the pandemic is one of a number of projects that we work on in Solaravus. I am not an expert per se in epidemiology, and I am not an expert per se in immunology, and so therefore the ideas that I'll be speaking about come from an educated biophysicist, but they would need to be taken forward to give them more credibility and to flesh them out by experts in those other areas. So the work was inspired by what I would call puzzling features of the pandemic data. And so what we're going to do now is turn to some slides that I've prepared that show various data from the pandemic, and I will talk you through those, and that will set the backdrop for the ideas that follow. What we see here is epidemic data taken from the website worldometers.info. They collect data from the governments or health services of every country that is suffering in the pandemic and post it each day. Data we're seeing right now is from the United Kingdom. And at the top we have new cases, and below we have the new deaths, that is the deaths recorded each day. Now the deaths are a much more robust measure of the impact of the pandemic, just because new cases are mostly quite severe cases which have required serious medical treatment and testing. There will be probably far more cases which are mild with relatively few symptoms or even asymptomatic, and those will not be captured statistically for most countries. The main thing I'd like to point out from this data is the fact that we see a very rapid increase in both cases and deaths. This is the very typical exponential nature of epidemic. We see some saturation, and then we see that the fall off of the epidemic is very slow, and it's that sluggishness of the decline of the epidemic that I really want to draw your attention to. We can look at another country. Here is Spain. You see very similar statistics, this exponential rise in cases and deaths, and then this very slow decline in the epidemic. And again, here's the United States, exponential increase, very slow decline in both cases and deaths. And I think a very natural question is, we understand the exponential increase. Of course, that's just a positive feedback of the initial infection. What's causing this very slow decline? And you might argue that that's something to do possibly with lockdown, that you have this kind of trickle of cases, so that the epidemic hasn't reached herd immunity and is sort of lingering in the population. Then, if that was true, you might expect that in a country that had not had really a serious lockdown, you would have a much sharper decrease in the epidemic. And if we look at the data from Sweden, we see that that's absolutely not the case. They too have this rapid increase at the beginning, which is the exponential increase of the infection, but then a very slow decline indeed. In fact, even with new cases starting to rise again. So it's not obvious that this dynamics is due to social behaviours like lockdown. So for now, just regard that as an interesting feature of the data that is quite robust across different countries. What we'll find is that the hypothesis I'll be offering provides an explanation for this. Next, I'd like to show you some data on cases and deaths as a fraction of population. So these are deaths per million of population. Again, we'll not look too closely at the cases because, as I say, that's much less robust data because there are many cases which will not be picked up because they are mild. So the thing to draw your attention to here is the extraordinary degree of heterogeneity in these numbers, covering more than 2 orders of magnitude. We see in some countries in Europe we have an impact of the disease that is over 600 per million. We have another country in Europe which had the epidemic at around the same time, which has a fifth as much mortality. And you could argue that in Germany they had much better protocols for testing and so on. But then we have in Eastern Europe these very low numbers, quite extraordinary, more than an order of magnitude less in the number of deaths. You might argue that perhaps they have different protocols there in how they sign death certificates. Maybe they're not testing for COVID. I think it would be hard, though, to account for more than a factor of 10 through just a different accounting system of death certificates. So a very interesting difference. Quite high death rate in the US, but then we look at Japan, which is 2 orders of magnitude less in the death toll, and Australia even less still. And Japan being actually quite close to the epicenter of the original epidemic in Wuhan in Hubei province in China. Brazil, very, very high rates of death, and as we know, as of late June 2020, this is actually still possibly on the increase in Brazil. Another puzzle, that great degree of heterogeneity between death rates in different countries. Is it due to differences in lockdown strategies or differences in genetics of populations? Maybe. Are we missing some very important idea? I'll be suggesting we are in the hypothesis that we'll discuss shortly. Even within one country, you see a great degree of heterogeneity that is not obviously linked to regions of high population density. In the United States, we have very, very high rates of death on the East Coast, but we have an order of magnitude less mortality rate in California, which is also a very densely populated state with huge metropolitan areas in cities like San Francisco and Los Angeles. Quite hard to explain. Another West Coast state with similar degree of mortality, despite being far less highly populated. In the Midwest, it's sort of in between the East and West Coasts. In a southern state like Texas, a very, very low rate compared to other states. Louisiana, much higher. This has been suggested to be due to the Mardi Gras as a super spreader event. And then Florida, which you might expect to be particularly badly hit given the disease tends to really target in terms of severe outcomes, elderly people, and Florida has a quite significant population of retirees. You might expect it to be one of the worst hit states, but indeed it's not. It has an order of magnitude less deaths per million than those eastern seaboard states. More puzzles. Interesting. And then the final data to show you is what's called zero prevalence data. These are studies that are done sampling randomly in the population looking for antibodies, which would indicate the infection rate of the disease. And somewhat surprisingly, even by late April, you see rates of infection that are below 10%, nowhere near what you would require for herd immunity, which would be up towards 70% infection. Indeed, for Sweden, this data was taken, I think, right at the end of April, and they have not been following a lockdown strategy per se. Still mysteriously low rates of infection, less than 10%. A very interesting study from Moran et al. Here in the group of Carl Friston, who've been using essentially sophisticated statistical algorithms, which are not models, but they essentially look for the most likely explanation for data using Bayesian inference. And they find that the most likely explanation of the epidemic data is that only a fraction of the population is actually susceptible. And they even indicate that that could be as low as 20% of susceptibles in the initial population. Very mysterious. And why would that be? Is it genetics? Is it diet? Is it lifestyle? Many people have put forward ideas of some kind of extrinsic factor that could cause this. As we'll see, the hypothesis that I will offer in this presentation gives a direct explanation of this, and it's intrinsic. It actually arises from the epidemic itself and doesn't depend on any other factors, and therefore would be seen to be true in every country. And indeed, this 20% figure is something that Moran et al. Find for 7 different countries in Europe. So it seems to be a robust figure, and that's consonant with what we'll see from the hypothesis to be discussed very shortly, which is intrinsic to the epidemic. So that's a range of data. Of course, there's far more data out there, but I just wanted to show you things from the pandemic that I, and I'm sure many other people, have found rather puzzling. Those long tails of the epidemic, the slow decline, the tremendous degree of heterogeneity in mortality from one country to another, and even within one country from region to region, and the low rates of apparent infection. How do we think about this? How do we explain this? So that was a sort of quick tour of a subset of the pandemic data, which shows a number of interesting features, perhaps some of those features a bit counterintuitive. Now, before I talk about the hypothesis of this work, I do want to just give a little bit of context from, a slightly more philosophical point of view, but this is important. When you're dealing with a complex problem, really in life, but particularly in science, there are 2 broad approaches that you can take. Let's call them bottom-up and top-down. Now, the bottom-up approach, which you might also call a reductionist approach, is based on the following premise, that, okay, we have some puzzling things that we're trying to understand, but we actually have enough information to hand to be able to understand the data. It's just that we need to combine these various elements of understanding in quite possibly an intricate way. So regarding the pandemic, that bottom-up or reductionist approach would be to say, well, we know it's complicated, but we understand the virus, we understand the immune system response, but we know that there are complex demographics, you have different genetic sort of backgrounds of various populations, you have different age structures, population structures, you have different timings of lockdowns, different travel modes in different countries, and so on and so on. And the idea would be if we can combine all of those factors, those human factors together in just the right combination by having possibly hundreds of parameters that we can fit to the data, then we will get an understanding that is quantitative of what's going on. And there is nothing wrong with that approach. If you do believe that you have all of the important information to hand, then really that is the only approach that you can take to understanding the problem. The other approach, top-down, is to say there's something puzzling. We know a lot, but perhaps we're missing something very important. Perhaps there's an idea that is missing, and if we are able to grasp that idea, we will immediately get a broad qualitative understanding of the data. Things will, as it were, fall into place. And then including all of those other details, things like age structure and population densities and lockdowns and all of that sort of thing, that will sort of fine-tune our ability to fit the data, but essentially we'll have a qualitative handle on what's going on by having this additional concept that we hitherto lacked. So these bottom-up and top-down approaches are really very common in problem solving, both in life but particularly in science. Let me just give you a very quick example. Let's say that you are trying to understand the brain. So many people spend their lives trying to understand the brain. A bottom-up approach to that would be to say, well, we know the brain is essentially a large number of neurons that talk to each other through synaptic junctions. If we can understand a neuron cell and the synaptic junction in enough detail, then eventually we can put all of that information together and we will have an understanding of the brain. Probably quite an optimistic point of view, given that we're trying to understand things like memory and consciousness when we think about the brain. The top-down approach is to say, well, it's just dazzlingly complicated, but are there certain phenomena to do with the brain that we can maybe capture at a high level? And that starts to be more in the field of behavioral psychology, where you essentially probe the brain through interacting with human subjects and you note their responses. And that gives you sort of a top-down perspective on some of the ways that the brain functions. In the ideal world, and this is really what we strive for in science, if science is done well, is to connect the bottom-up and the top-down. Neither is going to give you a satisfactory account of the phenomenon, but together, if you can join them, you have then a very powerful way to understand how to connect sort of mechanistic aspects, like in the case of the brain, the neurons, with behavioral aspects, which are perhaps more relevant to the everyday application of that piece of science. So it is with the pandemic. There are many people, epidemiologists, who are taking that reductionist bottom-up approach, and my job as a theoretical physicist who's interested in this is not to get in their way by doing more of that reductionist bottom-up stuff, but maybe to take a different point of view and say, oh, here's a new concept which might explain much of the data at a sort of a broad brushstroke scale, and maybe that can be really helpful from a top-down point of view. So, enough philosophy, let's now get down to brass tacks. The hypothesis, the idea that struck me very forcefully about a month ago, was that given in biology we know so many examples of what I would call population responses to perturbations from the environment, is there a population response to the pandemic at a biological level that is occurring? In slightly fancy language, one would call this an emergent superorganismal immunological response. Emergent in the sense that the response comes from the pandemic itself. Superorganismal really meaning at a population level. And then immunological response. And the hypothesis then is that there is some causal influence that connects populations of infected individuals with populations of susceptible or uninfected individuals, beyond the thing that we already know, of course, which is that contact between the two leads to infection of susceptible individuals. And so the hypothesis is that there is a more population level interaction going on beyond just contact between individuals, and that interaction is actually protective. As we'll see, this idea of a protective interaction can explain much of the data. The influence that mediates between infected and susceptible individuals I call a preconditioning field. So preconditioning in the sense of buffering or giving some degree of protection to susceptible individuals. So that's the hypothesis, that there is a population level preconditioning field that relates or connects infected populations with uninfected populations, potentially over quite large distances. That may seem quite vague. I'm going to be talking about a particular example of what that preconditioning field might be. I'll give the example of viral detritus, namely the fact that infected individuals won't just be breathing out live virus, they'll also be breathing out dead virus, viral fragments, viral detritus. That detritus will be structurally intact in the form of protein molecules and capsid fragments for very long periods of time in air currents around the world. And we can actually calculate what concentration of that viral detritus will be in the air all around us, which uninfected individuals will be breathing in all the time. And that's an example of this idea of a preconditioning field that connects uninfected populations with uninfected populations with infected populations. So we're going to go back to some slides now where I will talk in quite some detail about how that preconditioning field idea, I'm going to call it PCF, just as an acronym for preconditioning field, how the PCF idea can be very easily embedded in the normal epidemiological frameworks. And then we'll talk about some of the consequences of that, and you'll start to see how this does lead to a very natural explanation of much of that puzzling data. So I'll see you back in person in a wee while. In this slide, I'm trying to show the preconditioning field hypothesis in its very simplest conceptual form. I'm embedding it in the simplest epidemic model, which is the so-called SIR model, where SIR stands for susceptible, infected, and recovered. And I should just say right at the outset that even though here I'm showing what's very intuitive, namely that outcomes of infection can either be recovery or death, mathematically and sort of algorithmically, because these are both outcomes of infection that play no subsequent part in the infection, we are not going to distinguish them, and so we just call them recovered for short. And that's why we just talk about SIR. The way this works is that you divide your population into groups that are susceptible, they've never seen the infection. There's a population that is currently infected, and then there's a population which has had the infection. And the idea is that if a susceptible individual is in contact with an infected individual, there'll be a short-range infection process, and that susceptible individual will move across into this sort of category of infected. That model has been around for nearly 100 years, has been studied in great detail, of course, in epidemiology, and there will be hundreds of extensions of that model to try to take into account more realistic features of various epidemics and infections. Now let's put the preconditioning field hypothesis into this conceptual framework. First thing we need to do is to define another category of individuals, and we call them preconditioned. These individuals were susceptible, but through contact with the preconditioning field, they are no longer susceptible to becoming infected. How do they become preconditioned? Well, it's on sufficient exposure to this preconditioning field. That field itself is created by infected individuals. And this is a really important point about this hypothesis, that it is emergent. The preconditioning arises as an outcome of the state of infection. It's an intrinsic emergent property of the epidemic itself. It relies on no extrinsic factors whatsoever. So this makes it highly nonlinear. You can't have any preconditioning without there being infected individuals. So you have an initially completely susceptible population. Someone is infected, that infected person infects others, and you start to build up through positive feedback very rapidly an infected population. In the PCF hypothesis, they are releasing this preconditioning field, and that preconditioning field will get stronger and stronger the more infected individuals there are. The hypothesis is that on exposure to the preconditioning field, susceptible individuals can become preconditioned immunologically, which means that they're not susceptible to infection. We immediately see from here an explanation within the framework of this hypothesis of the Bayesian inference studies and the zero prevalence studies. The reason in this hypothesis that you do not see high rates of infection, and the reason why in Bayesian inference studies you infer a very low susceptible population even at the outset, is because of preconditioning. I should just add a very important point here. You would not expect there to be a large susceptible population as long as you have a preconditioning field from other regions. The very first region to experience the infection will not benefit from any preconditioning field at the outset, and so will have a much larger susceptible population according to this hypothesis. Now, we assume that this preconditioned state is not permanent. It's not something that is like the adaptive immune response that raises antibodies and gives you lifelong protection through memory T cells. It would be a transient state. Therefore, we would expect individuals to revert back to being susceptible as this preconditioning field weakens. And this explains within the hypothesis why you see such a sluggish decline in the epidemic curves. The reason for that is that as the infected population peaks and starts to reduce, and it starts to reduce because there are not enough susceptible individuals to keep feeding that infected population, as the infected population reduces, so does the preconditioned field reduce. And as the preconditioning field reduces, then there will be a reversion from the preconditioned state back to the susceptible state. That essentially replenishes the susceptible category, which essentially feeds the infection. And so you have a very nonlinear process here that we can't really understand in any detail intuitively. We have to use some kind of mathematics to look at the ramifications of this nonlinear process. But intuitively we can see how that can lead to a sort of a tug of war between the conditions of susceptibility and preconditioned, that infection would be able to reap from this replenished susceptible population through the reversion process. Later on, I'll show you the outcome of mathematical modelling of this conceptual model, and indeed we'll see very long, sluggish epidemic curves emerging from this nonlinear process. Let's just talk briefly about spatial aspects of this hypothesis. The idea came to me very much in a spatial context. I was thinking very hard about how can we have so much heterogeneity in mortality rates from one region of the world to another. And when the preconditioning hypothesis came into my head, it was very much in the sense of being a long-range influence. So if we think about how this might work, if you imagine the epicenter, which for COVID-19 would be Wuhan, and the surrounding province, then that epidemic will rage there at first. It will spread very, very quickly through local infections, short-range infections. But very rapidly too, according to the hypothesis, there will be a preconditioning field building up, and the preconditioning field is assumed to be longer range, and that will spread to neighbouring populations, preconditioning them, making them less susceptible to infection. So what we would expect is that as you go further away from the epicenter, as long as sufficient time has passed that there's been a degree of preconditioning, we would expect the infection, once it finally reaches there, to be less severe. We would expect less transmissibility of the infection, less cases per capita, and less mortality. Now, the preconditioning field is not thought to be instantaneous. It spreads, it has some time scale of spreading. Therefore, there will be regions very far from the epicenter where there will be no preconditioning for quite some time. That would not be a problem, except we have intercontinental air travel. And so the infection can be introduced into those very distant naive populations instantaneously, essentially by air travel. And I call those populations naive in the sense that they've not benefited from any preconditioning. Therefore, we would expect very severe disease outbreaks in those populations. Over time, these new epidemic populations will create their own preconditioning, and we would expect some, repeat of all of this nonlinearity in regions adjacent to these new epicenters. But initially, especially, they would have very severe disease outbreaks. And this picture does actually is consonant with some of that geographical data we discussed. The fact that regions that are on a continental scale adjacent to the original epicenter, so namely Eurasia, Japan, possibly Australasia, could be preconditioned and therefore expected to have weaker form of the epidemic. Regions further away, like Western Europe, may not benefit so much and would perhaps have more severe form of the epidemic. Those regions that are very, very farthest away, and if you look at the globe, the regions which are on the opposite side of the globe to Wuhan are the eastern seaboard of the United States and South America. Interestingly, that's where we see very vigorous, aggressive forms of the epidemic. So this preconditioning field idea as a sort of spreading influence that is not instantaneous does give, at first glance, a way of understanding that geographical heterogeneity. It is not the explanation, this is just a hypothesis, but it's interesting that it does give a plausible way that one could imagine how that great heterogeneity arose. This can be tested. Earlier I talked about one instantiation of the preconditioning field, namely viral detritus. That would be carried on global air currents. And it would be possible to look at air current data over the period January to June 2020 to see if regions of the world that experienced milder forms of the epidemic were essentially downstream in a causal sense through air currents from Wuhan province and other parts of the world which experienced very strong epidemic. Likewise, we would expect regions which experienced a strong epidemic to be essentially quiescent, to be not causally connected by air currents to the original site of the epidemic. And one can ask if that's true for Western Europe, possibly because of air currents from the Atlantic, and whether that's true for the eastern seaboard or for Brazil. I am not a geographer or an atmospheric scientist by trade, so I cannot myself give an immediate answer to that question, but those people absolutely will be able to inform that discussion. So that would be a very simple, in principle, test of the hypothesis of the viral detritus as a instantiation of the preconditioning field. So, we saw there in those slides that embedding the PCF, the preconditioning field, into the conventional SIR epidemiology framework is very straightforward. We were able to talk through the consequences of that in a sort of an intuitive way, and we see that yes, this idea can explain a number of features of the data. We talked about spatial aspects of the PCF, and again, we saw that there are some intriguing connections between the intuitive outcomes of the PCF and what we see in the data at a larger scale in terms of those very diverse mortality rates geographically. Now I want to speak in more detail about a particular instance of the PCF, an example. I think in physics we just temperamentally are more comfortable having abstract ideas, such as the wave function in quantum mechanics or the gravitational field that Sir Isaac Newton sort of came up with in the late 1600s. In biology, in my experience, scientists are a bit more hard-headed. They do like very specific instantiations of ideas, otherwise they'll tend not to really want to think about them very much. So it's beholden on me to give a plausible example of this preconditioning field, which I'm now going to do. So let's think about this idea of viral detritus. Infected individuals will be coughing for a number of days, and we know that part of that coughing they will be putting into the atmosphere around them live virus, and that makes these individuals infective. But it's not just live virus that they will be coughing up. They will also be coughing up essentially a mixture of other things. There will be fragments of virus, there will be dead virus sort of particles. There will also be immune system fragments as well. I call all of that viral detritus. A lot of it will be molecular. Now the point is that the virus itself needs to be in a very special environment to remain infective, to remain alive, as we say, even though it's not really what one would call an organism. However, the fragments of virus are not infective themselves, and they will remain structurally intact for potentially quite long periods. Thinking of just pieces of capsid of the virus or individual protein molecules from the virus, these molecules will remain intact for long periods and be in air currents. So we don't have to think about local contact when we think about viral detritus. So we need to start thinking about, well, okay, long distance transport in air currents, but surely the concentrations will be so tiny that this is not really a plausible way that we can causally connect infected populations with susceptible populations. And that's a very reasonable, intuitive response to this idea. But I believe it's incorrect. And the reason I believe it's incorrect is that I know that intuitive responses to these types of questions are generally very poor, because our human brains are not very good at estimating the product of a very large number with a very small number. The very large number is the number of viral proteins that an infected individual will cough up. The very small number is the concentration of those particles in the Earth's atmosphere. It's quite hard actually to intuitively, without really calculating something, work out the product of those two things, which starts to give you an idea of the probability of breathing in such things a long distance away. So there's a lot of detail on this estimation in the paper, and I won't go into details here, but let me just give you very quickly a sense of the numbers. And it all comes down to a beautiful idea that I first heard maybe 20 years ago, which at first sounds unbelievable, but you can calculate it very easily, and it really is the case. You have this rather daft question. You ask, every time we breathe, any of us, how many molecules do we inhale that were exhaled in the last breath of Julius Caesar? Et Brutus, you'll remember that gentleman was assassinated in March 44 BC. He exhaled one last time a number of molecules, air molecules, and the question is, of those molecules he exhaled, how many do we breathe in with every breath that we take? The answer is, with every breath we take, on average, we inhale 1 or 2 molecules from Julius Caesar's last breath. It's an amazing thing, and it comes from the following, that the effective volume of a molecule in a breath compared to the size of the breath, the volume of the breath, is almost exactly the same as the relative size of a volume of breath to the volume of the Earth's atmosphere. So when you combine those two numbers, you get essentially a number of order 1 as the product, and that means that we inhale on average about 1 or 2 molecules of Caesar's last breath every time we breathe. It's an amazing idea. We're much more connected than we would like to think to each other. So using that Julius Caesar sort of paradigm, it's actually quite straightforward to use the atmospheric aspects. What's much harder is to estimate, well, how many viral fragments are people coughing out or breathing out over the course of their illness, and one does have to make some crude estimates for that. So the numbers I'm about to give are within, I would say, 1 or 2 orders of magnitude. These are not precise estimates, but they can be made more precise by just doing experiments on viral detritus from infected individuals. But the best estimate is that an infected individual over the course of their illness will breathe out about 10 to the power of 14 viral fragments at the molecular scale. That's 1 with 14 zeros following it, which sounds like a large number, but with each breath we breathe out 10 to the power of 22 air molecules. So that's a 1 with 22 zeros. These numbers are truly enormous. If you have 1 million infected individuals in the world, the total amount of viral detritus that has been emitted into the atmosphere is 10 to the 20 viral fragments. If you assume that's uniformly spread over the globe, you can then work out that each of us inhales a viral fragment on average every 10th breath. That works out to be about 1,500 fragments per day that we would inhale, or 50,000 fragments per month that each of us inhales on average. And again, these are crude estimates. In reality, there's not enough time for that viral detritus to be well mixed in the Earth's atmosphere over the timescale of the pandemic. So now we start to see something quite interesting, that in those regions of the world that are downstream of air currents from heavily infected areas, like the original Wuhan epicenter, you will have concentrations of detritus that are hundreds or thousands of times higher than the estimate I've just given. In other areas of the world which are quiescent, that are not connected to viral epicenters through air currents, they will have essentially zero concentrations of viral detritus. And so if viral detritus inhalation is a preconditioning mechanism, if it's a PCF, then you will start to see geographically, you'll have regions of the world that are quite protected, that will experience very mild forms of the epidemic, and you will have regions of the world that are completely unprotected, and when they have the virus through long-distance air travel suddenly appearing in their midst in the infectious form, they will have no preconditioning and will have a sort of epidemic dynamic very much like the original Wuhan epidemic in a completely naive, unprotected population. So this idea is actually quite plausible and holds together relatively well. Another aspect that we must think about is playing devil's advocate. So what? Even if I breathe in 50,000 viral fragments over the course of a month, that's still a relatively small amount. It's mixed with millions of other pieces of detritus that I'm breathing in from many other organisms. Why would that give me any protection? Why would my immune system respond to that? And that's an excellent and important question. The idea, I think, is the following, that we know, it's been well documented, that when people receive vaccines, which really are a very similar idea actually, we inject either dead virus or altered virus into people to protect them from live virus. Injecting a vaccine does not create just a specific immune response to that particular pathogen. It actually creates rather broad-based immune response, both in the innate and the adaptive immune systems. And you find that people actually have quite strong immunological robustness to many other infectious agents for a period after the vaccination. Those papers are cited in the Solaravus technical report. So the idea is that in breathing in these COVID-19, SARS-CoV-2 viral fragments, that it triggers a low-level innate immune response that provides protection if the individual then has later contact with the live virus. One can think about that in a bit more detail. One can say that what makes those particular viral particles, fragments, from SARS-CoV-2 really important is that they are evolved to interact very strongly with lung tissue. So, for example, those spike proteins in the viral capsid of SARS-CoV-2 would presumably bind very strongly to cell membrane proteins in the lung epithelium, and this would be a very different form of interaction with the other viral detritus that one is breathing in, and one can easily imagine that that would trigger a low-level innate immune response. It's not dangerous, but it's still something that the body wants to deal with because it's a very strong biological interaction between a foreign agent and the host tissue. So that's the idea. It can also be thought about at a more local level, not just geographically sort of intercontinental air currents, but also, in a house. If you have an infected individual in a household, as long as uninfected individuals in the household maintain very good hygiene, they may not have contact infection with live virus, but they will be breathing in viral detritus all the time. We would then assume that they would build up preconditioning and be less likely to catch the disease. And this actually chimes with data from Germany, where very detailed studies showed that there was a surprisingly low level of secondary infection in households. And the PCF idea through viral detritus gives a very neat explanation for that. A less positive aspect of this idea is that for the immunocompromised and the elderly, this is sort of like a double whammy, because their immune systems being weaker, they probably can't mount a preconditioning response to the detritus. So they're not really preconditioned, and in addition, they will be susceptible, as we know, to severe forms of illness if they do contract the live virus because of not being able to mount strong immune responses. So the elderly and the immunocompromised do seem to get a double whammy from this, and that might explain some of the very, very severe mortality statistics we see with age or with pre-existing conditions. So that's discussion of the viral detritus instantiation. It may not be actually the only way we can biophysically envisage a PCF, and there may be other ways in which infected populations can interact with uninfected populations over long distances. I'm now going to go back to slides, and we're going to look at a numerical analysis of the PCF in the epidemiology model. I'll show you some equations, and we will very quickly look at what happens when you integrate those equations numerically, and we'll see that we do indeed quantitatively capture many of those pandemic data, which gives us proof of principle that yes, the idea not only intuitively makes sense, but when you run the equations, you indeed quantitatively find that you easily reproduce the features in the data. I'm going to speak briefly about a quantitative mathematical analysis of the model. There's much more detail about this in the paper, but this is really just to give you a sense that one can do quantitative analysis. So here's the conceptual model that I discussed earlier, and here are the mathematical equations that correspond to that. So very briefly, the terms and equations in black are the original SIR model, and the blue terms and equations here correspond to the additional aspects that one adds because of the preconditioning field hypothesis. We call the preconditioning field phi, and it has a dynamic, so it's created by infected individuals, and there is some environmental degradation of that field with some rate. And we have preconditioned individuals. The proportion of those is denoted by P. Their number increases through exposure of susceptible individuals to the preconditioning field, but they can also be lost to this reversion process that we talked about. So that's actually relatively straightforward. We can't solve these equations mathematically. They're nonlinear. They're too hard to solve. We can code them into a computer and get the computer to numerically solve these equations for us for given values of these parameters. I'm only going to show you the results from varying alpha and epsilon because it's just a preliminary analysis of this model, and in fact it would be quite interesting to do a more thorough, detailed analysis of this model in due course. So in the next slide I'll show you what happens when you vary the rate of preconditioning, and after that I'll show you what happens when you vary the rate of reversion. So we start by assuming no reversion, so that parameter epsilon is 0. So preconditioned is a permanent state. In the absence of any process to create preconditioned individuals, we just have the classic SIR model, and I've just shown that here as a control. So you have the infection growing and then decaying again. You have an increase in the recovered individuals and a decrease in susceptible individuals until you have herd immunity. That's classic SIR epidemiology. Now, adding in a little bit of preconditioning, we see now that as the infected population grows, the preconditioned population grows, because not shown here is the preconditioning field that has essentially created a competing process for susceptible individuals, not just creating infected but also preconditioned, and that preconditioned population grows. For this particular parameter value, it grew to about 30% of the population. And just to show that as we increase the strength or the rate of preconditioning, we see that that preconditioned population gets larger and larger until at this particular rate here, it's dominating the disease. So here 70% of individuals become preconditioned early on in the epidemic and are buffered from infection. So this is a proof of principle that the model for parameter values that are not tuned shows very strong buffering. And that is an explanation within the PCF hypothesis for those zero prevalence studies that show a very low rate of infection in the population. And it also explains the work of the Friston group of Bayesian inference studies that show that the data, the real world data, is best explained by assuming a very low level of susceptible individuals. And so that buffering is seen very clearly here through the preconditioning field hypothesis. Let's now look at what happens when we add in this process of reversion. On the right hand side here, I'm showing a epidemic curve, what you might call the shape of the epidemic. This is the curve that we were trying to flatten in different countries around the world through lockdown and social distancing. I think what I've chosen here for this curve is the rate of the change of this recovered population. So that could, for example, be the rate of new deaths. So it's essentially a shape of the epidemic, and is the same data that we looked at right at the beginning from worldometers.info for new mortalities each day. Now, with a very high rate of reversion, extremely high rate of reversion, this is just a test, because if reversion is so rapid that preconditioning doesn't last very long, we would expect preconditioning to have no effect at all on the SIR dynamics. And that's what we find here, that the preconditioned population never really gets started, and we just have classic SIR dynamics. And the epidemic curve is what we would expect. It increases exponentially and then it decreases exponentially once you have herd immunity. Now, very interestingly, when we reduce that rate of reversion down into the range of values that the other rate constants have in these simulations, and all of that detail is in the paper that you can find in the link below the video, very interesting thing happens. You see that this epidemic curve starts to get a very long tail. Now, we argued intuitively that such a thing was possible because of a drip feed back into the susceptible pool from the preconditioned pool. But here we're actually showing it much more formally through integrating these differential equations of the model. And indeed, the model does give this very long tail. And to me, this is actually very strong evidence that the hypothesis has great interest. It doesn't prove that it's correct, but it says, look, it's really explaining a lot of the data at this point. We need to take it more seriously and to think maybe it's capturing something that we hitherto were not taking into account, and it could be a very important factor in understanding the pandemic dynamics. If we reduce the rate of reversion still further, that tail sort of goes away, but it's then followed by subsequent waves of pandemic. And what's happening there essentially is that the rate at which preconditioned individuals revert back to being susceptible is so slow that the epidemic's kind of over by the time they get back to being susceptible. But there's still enough infected individuals around to basically trigger a new epidemic. And so you essentially start off with another epidemic. It just gets, it's much smaller than the previous one, however. And then it gets successively smaller in these waves. And this is all an intrinsic feature of the model. These subsequent waves are smaller because you're constantly building up in the background a truly immune population of recovereds. So if the PCF hypothesis is correct, and we are asking urgently about second waves of pandemic, then the PCF hypothesis will be very useful in allowing us a way to understand how second waves might arise. And it will be really in understanding the rate of reversion from preconditioned back to susceptible. This final pair of figures just is a control. It just shows if we remove reversion, we go back to the kind of dynamics we saw a couple of slides ago, where we saw a dominance of preconditioning in the buffered population. But notice that the epidemic curve, again, is very symmetrical here and has no tail. So this says to me that because we see these long, sluggish tails of the epidemic curve in the real-world data, that if, and I'm always trying to be very careful with these caveats, if the preconditioning field hypothesis is correct, then it's almost certain that there's a high degree of reversion going on, which is causing those long tails. You still have buffering, it's just that the buffering is much more dynamic. These data, I think, show proof of principle that the PCF hypothesis explains a lot of the real-world data, but it's explaining it qualitatively. We're not trying to fit this model to the real-world data. What we need to do as a next step is to embed this PCF mechanism into existing, quite sophisticated epidemiology codes. Now, I'm not an epidemiologist, so I don't have access to those codes, but groups who do use those codes on a day-to-day basis, if they're convinced that the PCF hypothesis has a degree of plausibility, would benefit from incorporating the mechanism into their codes, and then, because they have age structure and pre-existing condition structure and spatial structure in their models, they'll be able to do a good job of fitting their models to real-world data with the PCF mechanism built in. And if the PCF hypothesis is correct, what they should find from doing that is that they very rapidly find excellent fits to real-world data, that this mechanism in a way does most of the hard work in fitting the data. It kind of provides that missing link, and then finding parameter values of all of those other sort of bottom-up factors that we talked about earlier on will be quite straightforward. So that's the sort of more nerdy output from running the equations and looking at sort of computer output, but it's very reassuring that quantitatively our intuition is backed up by mathematics, and indeed this mechanism is capable of reproducing many of those features. So I'm going to wrap up now. I'm going to give a brief summary of what we've talked about, and then I want to just discuss briefly some evolutionary aspects of this and also some possible technological aspects of the idea of the PCF. So in summary, we found that this hypothesis of a preconditioning field that could connect infected with uninfected populations over possibly quite large distances is able to provide quite a robust qualitative explanation for many of the features of the COVID-19 pandemic. It gives a natural explanation for why we see low rates of infection in populations despite the epidemic having been in those countries for quite some time. This buffering idea explains that. It explains why the epidemic takes a long while to die down because of this reversion idea that if the preconditioning dies away with time, because it's possibly an innate immune response rather than an adaptive immune response, then you're continually drip feeding individuals back into the susceptible pool, and that leads to a very sluggish decline of the epidemic. It explains also some of the geographical aspects of the disease, particularly if we talk about the viral detritus instantiation of the PCF. You start to get an interesting hypothesis for why certain regions of the world have very severe forms of epidemic. Other regions seem to have quite mild forms of the epidemic. What's powerful, I think, in these explanations is that it doesn't require anything beyond the hypothesis itself. We're not relying on anything to do with particular genetic backgrounds of populations or age structures or population structures. It is emergent in the sense that with this hypothesis, all of the dynamics we've talked about comes from the disease itself. We talked in some detail about this viral detritus example of a preconditioning field. Surprisingly, it seems quite plausible, because at first glance you would think that the concentrations would be too low, but that's just our human intuition failing us. In fact, concentrations are not negligible, and it's for immunologists ultimately to determine whether it's possible that the immune system can be specific and sensitive enough to mount a sort of low-level immune response to SARS-CoV-2 viral detritus at low concentrations. So I hope you found that interesting, that hypothesis and what comes from it. It may all be wrong, but we won't know unless we test it. But what I would say is that through conceptual reasoning and through mathematical reasoning, it passes the first proof of principle tests, and that makes it a viable hypothesis that deserves to be looked at in more detail by the rest of the scientific community. And I am quite confident that that will happen if people are convinced that it's interesting and plausible. But let's just think now a little bit before the end of this presentation about evolution. We see around us in biology amazing phenomena that are clearly population level, superorganismal behaviors of organisms. The whole biology of social insects is an example of that. The self-organization of an ant nest is one of the great marvels of nature. There are other examples. People have studied the response of bacterial populations to antibiotics, where you see beautiful population responses. People have studied something called Dictyostelium, which is an amoeba which has a wonderful life history based on a population response, including altruistic behavior of some amoeba to others. So we know that there are many examples in nature of these superorganismal responses. And so the question is, well, are humans capable of that beyond what we might call behavioral or sociological population responses? So we're not talking about the fact that we can all run into our homes and have lockdowns. That's not the kind of response I'm talking about. Is there a more fundamental biological population response, given that we know other organisms do have such things? And this raises sort of an interesting question, because the immune system of humans is extraordinarily complex and is the result of hundreds of millions of years of evolution. And one starts to think, well, if evolution's gone to all the trouble, so to speak, given that evolution is blind, but let's, so to speak, say if evolution has gone to all the trouble of creating this incredibly sophisticated immune system for each individual, why wouldn't it let those immune systems talk to each other? If you have a new pathogen, why does every immune system have to do all the work de novo to tackle that pathogen? Why can't there be some level of immune response at the superorganismal level, some level essentially of communication between immune systems that allows infected individuals to give some degree of preconditioning to uninfected individuals? We've talked about viral detritus as a possible mechanism for that, but I think from an evolutionary point of view, given that we know superorganismal responses are very common in nature, it would seem odd that there is not a superorganismal immunological response in humans. And that starts to be quite interesting, actually, because you might then think, well, that means that every time we breathe out or breathe in, we're actually connecting human individuals together in quite non-trivial ways. You could sort of imagine that the cough response that we have when we have a flu or a cold, which you might think is a clever innovation of the virus to make us cough and spread it more, is actually more of a superorganismal immunological response, which is to say, I'm ill, I don't want people around me to get ill. This is now, anthropomorphizing a biological response here, but that the coughing essentially throws that viral detritus into the atmosphere and allows preconditioning, making everyone around the infected individual less likely, in fact, to contract the infection. So, from an evolutionary point of view, it seems actually quite interesting, and it touches on quite controversial areas of biology to do with the degree to which individuals in a species are connected. So it's sometimes called sociobiology. Edward Wilson was an early proponent of that. Rupert Sheldrake has had ideas in what he calls Morphic Resonance, which is a very controversial way of thinking about this, but still very, very interesting. Speaking finally about technology, if you have an idea in science that helps you understand something, we humans like to then innovate with that. Can we technologically enhance the preconditioning field? If it exists. Now, of course, we can only enhance it if we know what it is. We have to identify it. If it is viral detritus, then one can actually imagine quite easy ways to use that idea. We would essentially want to take viral detritus and make the concentrations higher so that everyone is very quickly breathing in that preconditioning agent. I sort of make a joke in the paper about wind farms not harvesting energy from the wind, but actually being used to pump out vast amounts of viral detritus onto unwitting populations, thereby giving them a degree of preconditioning. And maybe we'll be encouraging people to cough more, not less, although not near other people, because you don't want the live virus to be too frequently encountered. So these are slightly tongue-in-cheek suggestions, but you can see that there are quite nice technological aspects that one can very easily think about with this viral detritus instantiation of the preconditioning field. So that's that. I'm just going to show one more slide that just summarizes the title of the paper and the group of scientists that have very kindly given me some feedback. And also, actually, I will mention a less pleasant aspect that I've experienced in recent days. I just want to mention here the individuals to whom I've sent the paper. And some of these are colleagues of mine over many years who I sent the paper to just because I know they would be interested. Others are individuals that I do not personally know but who are experts in epidemiology, and the paper has been sent to them for their interest. Many of these individuals have provided me with feedback already, and I'm very, very grateful to all those that have taken the time to look at the paper and provide me with feedback. I do need to give a less positive statement though, which is that the paper has been sent to archives. Now, archives are publicly funded entities, generally publicly funded, which exist to act as repositories for preprints. So these are scientific results that have not gone through the peer review process yet and been published, because that process can take months or even years. And the archives exist to give rapid exposure to new science, because that helps everyone then move science forward, particularly in times of pandemic when we need to get ideas publicly spread as quickly as possible. I regret to say that all 4 of the archives there, bioarchive, medRxiv, the physics archive, and then a Swiss-based archive called preprints.org, have refused to host the paper discussed here on their websites. It has essentially been banned from those websites. I have been astonished by this. I have written many scientific articles over my career. I've never experienced anything like this. From my point of view, the paper that I have written is absolutely a scientific paper. It's a hypothesis which is then explored in a very logical fashion using theoretical techniques. And there are hundreds of thousands of papers every year written in exactly that spirit. However, these archives have denied public access to my work, and that's actually one of the reasons that I'm creating this YouTube video, because I want the public to know about this work, because I think it is interesting and it might be useful in the way that we combat the pandemic. Before ending, I would just like to give a great note of thanks to Rupert Sheldrake. I didn't know Rupert personally before this idea, but I have been in touch with him over the past month to get his feedback on the preconditioning field idea, given his years of experience with a similar concept that he has worked on called Morphic Resonance. Morphic Resonance is highly controversial, it's not accepted in mainstream science, but it is a very interesting way to think about the superorganismal behavioural responses of organisms. Rupert has been extremely generous with his time and his ideas and provided to me a number of very critical suggestions for the manuscript, which I have adopted. Thank you, Rupert. And I think there's an important, maybe ending message here, which is the following. Science is only strong and only positive if it is absolutely grounded on the free exchange of ideas and on those ideas being pursued in a rigorous way, both with logic and in a complementary fashion, testing the outputs of that logic against data. But we do need bottom-up and top-down approaches, and the increasingly dogmatic approaches and philosophies in science, which tend to be reductionist, will slowly but surely throttle and kill the scientific endeavour. And I think we already see the beginnings of that, and we must take action to liberate science from dogma and from monolithic approaches to understanding this beautiful world around us. So that's it. Thank you so much for your time. I think this has been quite a long video, and I really deeply appreciate your time in watching the whole thing. If you have feedback, please do send it to me, and I will make every effort to respond to everyone who gets in touch with me. So with that, I wish you well, God bless, and possibly see you next time.